Okie dokie. Okay, so let's get into, first of all, the parts of the immune system. And we can't have an immune system without the cells that make up the immune system. And those cells are the white blood cells, the leukocytes, right? And so, like the other blood cells, platelets and red blood cells, leukocytes are formed in the red bone marrow. Okay, they're going to be found there in the spongy bone. And we briefly talked about the different types of leukocytes. All right, we have our granulocytes, and those are going to be the cells that have those little specks inside. So anything that ends in a fill, all right, so neutrophils, eosinophils, and basophils. Then we have one of the coolest leukocytes, and those are the monocytes. In circulation in the blood, they are monocytes. When they enter into the tissue, all right, they become macrophages. All right, and then we know from chapter 5 that there's two types of macrophages. You have the resident macrophages that just stay stationary and hang out in the connective tissue. And then you have the wandering macrophages, and they can move around, okay? And then finally, we have our lymphocytes. And that includes our B lymphocytes, our T lymphocytes, and a really cool named cell, the natural killer cells, or the NK cells. So these are all the white blood cells that are being produced in our red bone marrow. All right, so we're going to actually see, interesting fact here, that most of our white blood cells are not in the blood, but in tissues, right? And so some of these secondary lymphatic structures, these tissues, all right, are going to be located all throughout the body. For example, these guys here, all right, are malts, and we'll talk more about that in a little bit more detail when we get into the digestive system. But you have these clusters, all right, of white blood cells just scattered along your digestive system. And of course, when you're taking material from the external environment and placing it into your body, there's a good chance that there could be a pathogen or two in there. So of course, we have these white blood cells, these clusters all throughout, that are going to get called into action. And there's a condition out there called leaky gut syndrome. And that's literally when you're on an inflammatory diet, and inflammation causes swelling and it makes things leaky, right? And so you'll see, all right, these malts are going to be highly active in folks that have a horrible, horrible, highly inflammatory diet. But we'll talk about that when we get to chapter 26. Okay, so you'll see here we have our secondary lymphatic structures, all right? We have all those, the T cells, the B cells, macrophages, the dendritic cells, which are a really cool classification of cells because they like to find pathogens and then show it. It's like that little kid that finds something on the ground and comes over to its mom and dad and is showing them what they found. Well, that's kind of like what these cells do. Then you get your natural killer cells. They just roll around. Anything abnormal, they kill. They're not specific. They just go after it. Dead, dead, dead. All right. So we'll see all those located in the lymph nodes. So if you ever wonder why you get sore lymph nodes when you have a virus or a cold, well, that's because all those cells are hanging out there and those lymph nodes become enlarged. All right. So we have our lymph nodes, spleen, tonsils, the malt, and the lymphatic nodules. Now we'll notice that throughout our body, the macrophages like to chill out in certain areas. One of the famous areas is in the lungs. And if you think about it, when you're inhaling the air coming from the external environment to the internal environment. So of course, there's pollutants in the air, right? Certain airborne pathogens, okay? For example, COVID could be, is considered an airborne pathogen. So when said person inhales the air, all right, these macrophages, if they come in contact with any pathogens, will initiate, all right, the immune response. In most cases, they'll just go over there and just gobble up whatever it is they find, all right? But we'll see they're just going to hang out and they stay there forever. They find a nice comfy home and they show up and they hang there. And then we have our ones that come and go, and they'll come in and leave, all right, because there is a blood supply to the lungs. All right, again, we'll talk more about that when we get to the respiratory system. And so we'll see, right? They're going to hang out there and say, hey, you know what? I like living here. I think I'm going to take a permanent residence here. And then they're going to do what they do and just keep an eye on what's going on. They're like the neighborhood watch, making sure that nothing bad is happening. Well, in chapter 6, and I just got done teaching that to my 210 class, we talked about, all right, the different layers of the skin and the cells that are found there. All right, so when we're dealing with the epidermis, all right, that's the epithelial layer of the skin, we talked about the cells of Langerhans, or the dendritic cells. All right, and these cells, all right, they're also located in our mucous membranes. And so think of mucous membranes being located in areas that are entrances and exits to your body. Okay, so you're going to see it, nose, throat area, mouth. All right, we're going to head down into the anus, the vagina. All right, you're going to see these mucosal membranes all throughout. And so they hold on to these dendritic cells. And so these dendritic cells, all right, are in a classification of what we call APCs, antigen presenting cells. And that's that cell I was telling you about, that they're going to just come across a pathogen. Again, they're very non-selective, all right, or non-specific, I should say. They just see something they don't recognize and they're like, well, you know what, I'm here, it's here, let me just take care of this thing. And that's what it does. It goes over there, it phagocytizes whatever it finds, and then it makes its way into the lymphatic system. Now, eventually the lymphatic system dumps everything back into the bloodstream. And then that goes through to the proper channels. It's going to run through, all right, the spleen, and that's where we see a lot of cleansing and cleaning of the blood, of course, in the liver. All right, but again, hey, I know I say this quite a bit. We'll talk about that soon. All right, so there are epithelial cells of the skin, okay, in the epidermis, and our mucosal membranes are going to have these specialized dendritic cells. Right? And their job is, when they come across something, a pathogen, is to basically just eat it up. And then we'll go through the process of what they do afterwards. Because basically they'll eat something up, they'll break it down, and then they just stick whatever it is that they just imbibed, and they put it all over its body, right? All over the cell. Right? It's interesting. We'll talk about that. All right, then our connective tissue, the most abundant type of tissue in the body, holds on to these famous fellows, the mast cells. All right. Now we'll have mast cells in our tissue, and there is some in circulation here. All right, but we'll see them close by areas that have vascularity, small blood vessels, right? And that's what they're going to be. They're going to be hanging out there because of their role. And we'll talk about their role. They're basically going to make blood vessels leaky, which is good because we want to get stuff out of the blood into the tissue if there's damage to the tissue. And they do a wonderful job with that, okay? So we'll see tons of the mast cells in the dermal layer of the skin, okay? The dermal layer primarily is made up of dense irregular connective tissue, and that's highly vascularized. So again, we'll see lots of mast cells around there. We'll also see it around the mucosa, right? So again, if you're having allergic reactions to things, you know, like seasonal allergies because your mast cells are kicking into overdrive, all right? Again, you'll see this in those tissues there, all right, along our respiratory system, the GI tract, hugely, hugely important, and then along the urogenital tracts. So consider that. You're going to see a lot of these mast cells in the different types of connective tissue throughout your body. And so this picture here is just displaying just some of the areas of where we're going to see these immune cells here. So here are our leukocytes, the various types in circulation. All right, we have our granulocytes there, and then you have your lymphocytes and your monocyte. Okay, and then in some of our secondary structures, all right, this is an example here. We're looking at a lymph node here. All right, and we'll go over the anatomy of the lymph node later when we get into the lymphatic system. All right, then you can see here we've got one of our resident cells here. The macrophage is just taking up its new home here in the lungs, constantly going to be monitoring what you're inhaling. Then, of course, our dendritic cell we'll see in the epidermis there, in the stratum spinosum there, okay, excuse me. And again, you can see how it's just kind of splayed out amongst these other cells here, these keratinocytes, and it's monitoring them, seeing if they're okay, if they're normal, if their cell receptors are normal. And if they start to produce irregular cell receptors, then it will immediately kill that cell. And then our mast cells, which are located throughout the various tissues of the body here, all right, we'll see them close to obviously blood vessels, right? But in this example here, you can see it in the muscle tissue. So in order to really understand the immune response, we've got to kind of understand how it communicates within its own system. So our immune system has a way of communicating, all right, to the various structures and players of the immune response. And that brings me into this term here called cytokines. And they're proteins, okay? And they're proteins that are produced, think of them as chemicals, all right? And they're going to help with governing and monitoring and regulating the immune response, right? So our immune system is made up of two parts. The first part is called the innate, and the second part is the adaptive immune system, okay? So we're gonna talk a lot about the innate today. The next class, we'll talk about the adaptive, all right? But these cells are in both. They're going to be found in both our innate and our adaptive. Now, the innate is basically what you're born with and you keep it. It's a very passive form of the immune system. For example, like your skin, that's part of the innate immune system. It's a covering, it's a protective covering. It keeps things outside of your body for the most part, okay? So the cells here, these immune cells, are going to release chemicals, all right, when they come in contact with something, all right? If they need to get a message out like, hey, you know, I think I found something here. I need help. I need more people, or excuse me, more cells to come over here and take care of, or at least help me out with this situation here. So whatever that cell is, it's going to release, all right, its cytokine, all right? And then it will flow around throughout either the lymphatic system, the tissue itself, or the bloodstream here, and it's going to look for the target cells. All right, basically, you know, saying, hey, come here, I need some help. Let's help me work on this. So when we talk about the effect of cytokines, there's three different ways, all right, that a cell can react to the release of cytokines. One, it can release it on itself, all right, and that is called an autocrine response, or it can release it within the close proximal vicinity, all right, that's called the paracrine, and that will work on the local cells there, or it can try to recruit cells from all over the body or in various regions that are far away, okay, but to do so, it has to circulate through the blood, and of course, we've already come in contact, all right, with endocrine, all right. Cytokines, yes, do have a short half-life, which means they don't last very long, all right? Because again, we don't want this to be an ongoing thing, all right? If you keep putting it, it's like driving your car. I mean, when you come home, all right, from the grocery store, you take the keys out of the car, all right? Turn the car off, take the keys out of the car because you're done with it, right? I mean, you're not going to leave it running all night until the next day when you have to go to work. You don't do that. Same thing here, all right, with the cytokines. All right, short half-life, we don't want this response going on and ongoing, okay? Okay, so some of the effects that we see here, all right, are going to be to signal cells, like I was saying. Hey, we need some help. We'll see that in some of our non-immune cells. The example here are the neurons, okay? The neurons, obviously, their job is to transmit, all right, nervous signal information, all right, to get some sort of response, right? And so that is going to be, which is an interesting fact because white blood cells have specific receptors for, all right, neuronal interface. So the functioning of your nervous system, as long as your nervous system is functioning pretty well, that will only help and improve your immune response. That's why it's always important to take good care of yourself, right? Very, very important. All right, the cytokines are going to do several, again, think of them as a chemical messenger, right? So they're going to help with the development of immune cells and how the immune cells are going to behave in how they react to the immune response, okay? Plays a big role, and we'll talk about this hopefully today, in the inflammatory response. And of course, all right, in the role of destroying cells. All right, so cytokines are an important player, all right, in our immune response here. All right, so let's look at, all right, innate immunity and adaptive immunity. All right, innate immunity and adaptive immunity. So a couple of the differences between innate and adaptive immunity is, one, what cells are involved? Okay, because when we get into adaptive immunity, we're going to talk about the cell-mediated response there. So that's going to be very important. All right, so what cells are going to be involved? All right, and the cells that are involved. All right, how specific? How are we going to get rid of whatever it is that invoked the immune response? Okay, if you have a virus or a bacteria, how do we get rid of that stuff? And then, of course, just like with most things, time. How long is this going to take? Is this going to be a couple minutes, a couple days, a couple weeks? Hopefully not months, okay? But it all depends on what is going on, okay? So there's going to be some really stark contrasts between innate and adaptive immunities. But keep in mind, all right, innate and adaptive immunity, their overall function and job is to protect the body. It is to protect the body from some sort of pathogenic response. So innate immunity, we call it innate because it is present at birth. You are born with innate immunity. All right. Most importantly is innate immunity is non-specific. So it's basically going, it doesn't show favoritism towards anything. Right, when we're dealing with non-specific, all right, for innate immunity, right, it is going to be a broad spectrum of protection. Right, and it doesn't matter if you've been exposed to the pathogen or not before. Right now, when we get to adaptive immunity, that's a different story. Right, and that kind of feeds us into vaccines and whatnot. A lot of what you've been hearing here. All right, so when we talk about innate immunity, like I said before, right, that includes the skin and the mucosal membranes. All right, and those are essentially barriers. That's what their job is. All right, so they non-specifically prevent infiltration, all right, of pathogens, things in the external environment, from getting into the more sensitive internal structures of your body. So we'll see a non-specific response, and we'll talk about what happens internally, all right, within the skin and the mucosal membranes here. The nice thing about innate immunity is that the response is immediate, okay? Your skin right now has tons of bacteria living on it, and we refer to some of that bacteria as opportunistic organisms, which means if they're presented the opportunity to cause an infection or to cause harm, they'll do it, okay? But thank goodness your skin keeps those organisms out and doesn't let them enter in. Now, adaptive immunity, okay, also known as acquired, this is specific. And so we're going to see how an antigen, now an antigen can be either a foreign protein or a foreign carbohydrate. All right, it's something that's foreign to your body, and your body recognizes it as what is called non-self. So you have self, which is you, okay, and then you have non-self, something that isn't of you. And if something is non-self, most likely you'll get an immune response to it. If it's self, that means, oh, it's supposed to be here, then your body leaves it alone. When I say your body, your immune system leaves it alone. Okay, so what we'll see here with adaptive immunity is we'll have an antigen, right, which is going to then mobilize and activate our T and B cells, or our T and B lymphocytes. And so we'll have a specific or a particular cell that is going to respond to a specific, that's important, specific foreign substance or an antigen. Now, this one takes a little bit longer. It can take several days, possibly even weeks. all right, for this to kind of kick into effect. And we'll talk about why that is and all that fun stuff. Okay, so what you're looking at here is a nice chart here for the immune system. All right, on the left side of the screen, you have the innate immune system. All right, and so we have, all right, of course, keep in mind, non-specific response. So skin and the mucosal membranes, again, preventing entry. Okay, and then we'll have also non-specific internal defenses. So if something does manage to make its way in, well, that's okay. Well, it might not be okay, but I'm just saying it's all good because you will still have a chemical reaction, all right, in regards to what cells are responding, all right, macrophages, natural killer cells, all right, and then some of the chemicals that are going to be produced, all right, by some of the structures here in the immune system. And then we'll talk about some of the physiological responses. We should get through all of that today. So then on our next class, we'll talk about our adaptive immune system. Here, we're going to talk about specific cells, T cells and B cells, and how they respond when they encounter a specific antigen. All right, and then we get to learn about here when we're talking about humoral immunity, all right, B lymphocytes, we're going to talk about the famous plasma cells and how they make and release antibodies. You hear about that a lot with COVID, all right, antibodies, antibodies, antibodies. So we'll get into that. So let's get into the specifics now of innate immunity, all right? Keep in mind that innate immunity is preventing things from getting into your body, all right? And it does so in a non-specific manner. So we liken it to the first line of defense. All right, and so that's going to primarily be the skin, largest organ in the body, okay, plays a protective barrier, and then the mucosal membranes, which will be found, all right, in and around those areas that are entrances and exits to our body. Now, say things do manage to make it past our first line of defense, no worries. We have our second line of defense, and that's going to be the internal processes, right? Which we're going to be dealing with certain cells, not all, but certain cells. And then we'll talk about how your body will produce certain chemicals, which will help with the immune response. And then we'll finish up talking about, all right, the wonderful process of inflammation. Inflammation is good. You want to have inflammation. All right, but I have to put a big but there, all right, that too much of anything can be bad. And that's where you start to run into issues with inflammation, if you get chronic inflammation. Right? But you want to have inflammation because it helps facilitate the healing process. That is important. All right, so how do we keep things out? All right, well, our skin. All right, and notice this is an important descriptor here. Intact skin, all right? Obviously, you don't want to have a cut, all right, or tear in the skin, all right? The skin needs to be intact. And so intact skin makes it very difficult for pathogens, all right, to enter in, all right? And if you recall, all right, our epidermis is epithelial tissue. Epithelial tissue, one of the characteristics of epithelial tissue is that it's tightly packed. Tightly packed. It's avascular. It's so tightly packed, we can't even fit blood vessels in it. All right, so our epidermis is tightly packed, and that makes it very difficult for these microbes, all right, to get into the inner part of that tissue there. In addition to that, how awesome is this? Our skin produces, all right, all of these antimicrobial substances. Right? We talked about, for example, sebum, right? Chapter 6, when we were talking about the sebaceous glands, which are holocrine glands, all right, they produce the sebum that is excreted onto your hair. And as the hair, all right, is passing from out of the hair follicle, you know, outside the skin, it deposits the sebum onto the skin. And so all of those antimicrobial substances listed there help to keep in check, all right, your microbe population on your skin. Because let's face it, there are tons of bacteria that live on your skin, and we refer to them as our normal non-pathogenic flora. And they just hang out there, right? And yes, they're microorganisms, and that includes, it's not just bacteria, right? But it also can include, all right, fungus, for example. And so you will see how with these antimicrobial substances, they help to keep, all right, this non-pathogenic flora in check, make sure that population does not get out of control. All right, so our mucous membranes, right, we will see them producing mucus. Now, mucus, for the most part, and we'll see it when we talk about the digestive system, I mean, it plays a huge role in decreasing friction. It's a lubricant, okay? We talked about certain pseudostratified, ciliated columnar epithelium. All right, you will also see ciliated simple columnar epithelium with the goblet cells. They produce mucus, and a lot of that mucus is used to help with reducing friction. All right, well, here we're using it almost like a net. Mucus will catch the pathogens, any of these microbes, all right, get it stuck in the mucus, and in the mucus, we have more antimicrobial substances. And this last guy here, that's an antibody, immunoglobulin A, IgA. You will find large amounts of IgA, all right, in your mucous membranes, all right, and it's a protein. And we'll talk about how antibodies work, okay? But again, we have these antimicrobial substances which are embedded into these mucous membranes here, and their job is going to be, again, to keep any pathogen or microbe that has managed to make it that far, its job is to limit and kill, all right, as much of that pathogen as possible. Okay. So that is our one of our, let me go back here to, oops, there it is. All right. So skin and mucosal membranes done. Now what we're going to do is we're going to talk about our non-specific internal defenses here. All right, non-specific internal defenses. So let's talk about cells that like to eat things. The Pac-Man cell, and those are our phagocytic cells. The neutrophils, the macrophages, and the dendritic cells. And their job is to wander around, all right, and not just eat microbes or anything that's considered to be pathogenic, all right? But for example, macrophages go around, if there's damaged tissue, all right, if you've injured yourself, they go around and they chomp up that damaged tissue. So our neutrophils and our macrophages, they go around utilizing phagocytosis, and they gobble things up. And so when they gobble these things up, they're going to pull it into the internal environment of the cell. And so they're actually going to make a vesicle. All right, and we covered that when we talked about phagocytosis way back in chapter 4. All right, and so it creates this vesicle, all right, that is closed off and isolated from the internal environment of the cell and the cytoplasm there. And so what happens is we get one of our wonderful lysosomes. Hope you can really appreciate these lysosomes because they're cool. And that comes over and it fuses with that vesicle. And so we call that a phagolysosome. And all those awesome digestive enzymes, all right, that were inside the lysosome spill out into that vesicle, all right, and they start to break down whatever is inside of that vesicle. And so we have this phenomenon called the respiratory burst. Now, this is huge because especially when we're dealing with certain microbes here, all right, this is a highly reactive oxygen-containing molecule process. So during that respiratory burst, all right, we're basically going to dump in these highly reactive, and when I say highly reactive oxygen-containing molecules, is these molecules, they are so, when I say volatile in a good way, they're just going to go and tear apart whatever they come in contact with. And it's a controlled environment there. So when the respiratory burst occurs, all right, these reactive oxygen-containing molecules will just go in there and destroy any microbes that cell has engulfed. And then it breaks up all of that, whatever it is, down into like amino acids and carbohydrates, and then it spits it out through the process of exocytosis. And that's what those neutrophils and macrophages do. I mean, they're heavy lifting cells. They do a great job. Now, the third cell is the dendritic cells, right? And it's, I don't know why, I think it's funny, but basically these are the cells that come in contact with something, okay? And they'll engulf it, all right? So basically, an easy way to describe the dendritic cells is they destroy particles and then they present the fragments. This is the one that I was telling you about, like that little kid that finds something and goes to show it to its parents, okay? So basically what will happen is the dendritic cells will come across whatever this particle is or this substance, it'll engulf it, all right? It'll break it down, and then it sticks, all right, the fragments all over the surface of itself, all right? So we refer to those fragments, all right, as antigens. Okay, so it's going to take those antigens and it's going to put it all over the surface of the cell on the plasma membrane, and then it's going to go look for T lymphocytes. And it's going to present those antigens to the T lymphocyte. And so this, and we'll talk about this later on, not today, all right, this will initiate the other immunity that we're going to be talking about, the adaptive immunity, all right? Cool thing is macrophages do the same thing. They can also do it, right? And so we call this classification of cells, the dendritic cells, the macrophages, those are what we call antigen-presenting cells. Antigen-presenting cells, because that's what they do. They'll take the antigen, put it on the surface there, go look for a T cell, and then see if it is something that can initiate, all right, the adaptive immunity response. Those are our dendritic cells. So that's what we're seeing here. Okay, so here's our dendritic cell, all right, and you'll see the whole process, all right, of here's some infectious agent, all right, we have our macrophage imbibing it, then it has a lysosome that fuses here with that vesicle, and it forms the phagolysosome, and then the digestive enzymes in that phagolysosome that came from the lysosome will break up, all right, that infectious agent through that respiratory burst. And so you have all those little particles there, and then it gets rid of it, just shoots it out. And then your body will clean that up later on. So that's what we're seeing here. Here's the macrophage doing its thing. And here, the dendritic cell, you can see all these little structures on the surface here. We'll talk about what those structures are, but that little ball there, looks like green or blue, right? That's the antigen that it took in, and then now it's covered itself with. And the macrophage could do the same thing. All right, let's talk about now our what we call pro-inflammatory chemical secreting cells. Pro-inflammatory, meaning we're encouraging inflammation. Right? Basophils and mast cells are very similar. Right? Yes, they both promote inflammation, that's obvious, but what we're going to see is the basophils are going to be the cells that are in circulation in the blood, and the mast cells are going to be residing in our connective tissue, right, in the mucosal membranes and some of the internal organs. Right, but they have very similar, all right, processes, meaning they both have all right, inside these cells, these granules. And again, if you look, basophils are granulocytes, but mast cells, when you look at them under the microscope, you can see granules also in those cells, right? And those granules have various chemicals, okay? And those chemicals are going to prove to play a major role in helping with the immune response. So we'll see when these cells release the granules and those chemicals, all right, they're going to have certain processes. Let's talk about a few of them. One, we'll see, all right, that these chemicals are going to help promote, all right, the movement of fluid from the blood to the injured tissue. Well, that's what we want. That's what we want. If you've damaged the tissue, say you've scraped your knee, okay, and that tissue is damaged, all right, we need to get stuff from the blood, because we already have some cells there in that tissue that are going to go to work right away. But again, we need more help. So to get that help from other places in the body, we have to rely on our circulatory system. And so these chemicals here, all right, are going to be released by these cells, all right, kind of like a carrion call, where we're going to be like, hey, we need help, come to this location. And we're going to send these chemicals out, and they're going to be what we call chemotaxic chemicals, which basically tells the immune cells, come to this area here. There is an issue going on. So chemotaxic chemicals attract other immune cells, right? So they can get to that area and help fix the problem. So histamine and heparin are going to be two of these chemicals that are going to help to promote inflammation. All right, one, they're going to increase vasodilation, and in doing so, they help to make the capillaries more leaky. So the stuff that's in the capillary can get into the tissue. Okay, so histamine is going to increase vasodilation and make the capillaries leaky, where at the same time, we have heparin being released so we don't clot the blood. Because if we clot the blood, then we can't get things into that area yet. So we don't want the blood clotting quite yet, right? We need to get a nice flow, all right, so we can get more immune cells to that area there. And so we'll see, you've seen this term before, the eicosanoids here, right? These eicosanoids are going to come from the plasma membrane. I don't expect you to remember this, all right, but there is a certain phospholipid in your plasma membrane called arachidonic acid. And that is the phospholipid that will start to break down and go through a series of chemical reactions, what we call an enzymatic cascade, and it is going to help produce prostaglandins, prostacyclins, leukotrienes, thromboxanes, all these locally acting hormones called eicosanoids. And they all start in the plasma membrane, meaning they come from arachidonic acid and basically goes through a series of chemical reactions which help to increase inflammation, which is what we want. All right, so here you can see the basophil releasing, all right, histamine and heparin. So we're making our capillaries and the arterioles larger, they're vasodilating. We're making them leaky, and we're preventing them from clotting. And then these basophils, all right, because of their plasma membrane, they got that arachidonic acid there, it starts to undergo the enzymatic cascade of creating these eicosanoids. And these various eicosanoids, the ones that I named, are also going to help to promote and increase inflammation. It's that positive feedback process going on. All right. How about our natural killer cells? Let's go back here to our little checklist here. All right. We're continuing on with the different cells here. So we talked about macrophages and their role. We talked about basophils and mast cells. Let's talk about some of our natural killer cells. It's just a cool name, natural killer cells, because that's what they do. They kill things. They go around and destroy any cell that is hurt, injured, irregular, okay, unhealthy, unwanted cells are going to be victimized by natural killer cells. Now you see a term up there called apoptosis. These apoptosis-initiating cells. Basically, apoptosis is cell death. So, and these cells are aptly named for this, okay? So like the rest of our immune cells, they come from the bone marrow, then get shot out into circulation. And, they'll circulate in the blood, and then they'll find our secondary lymphatic structures. So that could be the spleen, that could be lymph nodes, all right, MALT, different lymphatic tissues in which they'll just hang out there. And their job is to watch and surveil. That's what they do. All day long, every day. They just float through the body and they inspect things. And so they look at the cell to see if it has the right receptors being displayed. Because if a cell gets unhealthy, right, then it changes its appearance. Right? Just like if you're looking at a person, right, that is overall unhealthy, their skin might not look, might be yellow if they have jaundice, for example, right? But so the cells are no different, that when they start to become unhealthy, their appearance can vary. And these natural killer cells pick up on that. And so if a cell gets infected by bacteria or a virus, all right, or they undergo transformation, they become dysplastic, or even, God forbid, they become neoplastic, all right, and start to change into a tumor, all right, these natural killer cells, all right, can identify that for the most part, and they can then go to work on trying to kill those cells. How do they do that? They release these chemicals, cytotoxic chemicals, right? Cyto is cell, okay? So these are toxic chemicals to the cell. And there's two types. We have our perforin, right, which is really kind of cool, because what the perforin does is it creates holes in the cell. So these cytotoxic chemicals form perforins in the unwanted cell plasma membrane. And then what happens is, all right, the natural killer cell then injects granzymes through that membrane pore, and these granzymes kill the cell, all right, by causing that cell to shrivel up, right? Apoptosis, know that term, apoptosis, cell death, cell death. That's what happens here. I'll show you a picture, okay? So here's our natural killer cell. It's just floating around, doing its thing, checking on cells, making sure that they are where they should be, and making sure that they're healthy. Okay, so if it comes across a virus-infected cell and it notices, hey man, there's some weird receptors on the surface of that cell, I'm gonna go kill it. So what does it do? It's gonna release the perforin in the granzyme. The perforin makes those holes, the granzyme enters in, and then those granzymes end up killing the cell, all right, apoptosis, and causing that cell to shrivel up. Bye-bye. And that's our natural killer cell. Real simple. Now, we briefly talked about eosinophils when we were back in, not chapter 19, chapter 20, when we were talking about the circulatory system there. And our eosinophils, yes, we see those in high levels, all right, of people that have allergies or even asthma, okay? But really what we'll see, if you see on a CBC with differential, which is a blood draw in which they break down and look at the different cell types, okay? If you see an elevated eosinophil level, you're thinking allergic response, or this individual is undergoing a multicellular parasitic infection. All right, so very similar. But we're going to see that these cells, again, Phil is the last part of the name here for this type of cell. So it is a granulocyte, all right? So those granulocytes are going to be released, all right, into whatever it is the cell is attacking. And it does the same thing, creates those transmembrane pores, okay? And then we're going to dump in those toxic substances into those transmembrane pores to kill the parasite, right? We will also see these cells undergo phagocytosis. We'll talk about what the antigen-antibody complex is. But basically, that's when an antibody, all right, gloms onto an antigen, all right? And so they go around, they just kind of clean up that mess. Okay, so when we are talking about eosinophils, keep in mind, immune responses for allergic reactions and asthma or infections of multicellular parasites. All right, so last part here, and this is very important when we talk about, all right, cells of the innate immune system. All right, and I mentioned this before, these cells go around and look at, all right, our own body cells, but also anything that is not part of us or non-self, right? And it's able to do so, it's able to recognize those microbes, all right, as non-self because of the receptors. Again, if it doesn't recognize a receptor, it is going to kill it, all right? It only knows what it knows. And so it's better to be safe than sorry. So that's what they do, right? They're going to look at the cell surface receptors through pattern recognition. And if it's a different pattern, and that's usually what we'll see on microbe surfaces, well then guess what? We're going to bind to that cell and we're going to cause an issue for that foreign cell and we're going to kill it. So here you can see the eosinophil, all right, doing what it does best, releasing the cytotoxic chemicals, creating those transmembrane pores, and then pouring in, all right, those dangerous cytotoxic chemicals into whatever it is that it is attacking here. In this case, it's a parasitic worm, and destroying that worm. All right, so let's go back to our slide. Now, we just finished cells. So now we're going to talk about chemicals for our non-specific internal defenses. Chemicals. So let's talk about a few. All right. So we have our antimicrobial proteins. And look at the term. It tells you exactly what it is. These are proteins. These molecules are going to function to destroy, attack, what have you, microbes. So the first classification is what we call interferons, right? Their job is to stop the spread of viruses. Here again, it is a non-specific release, and that's what cytokines are. They're going to be non-specific, but their job is to impede, stop spread of viruses. So there's a couple different interferons that we're going to talk about. We're going to talk about interferon alpha, interferon beta, and then the gamma interferon. Right? So the alpha and beta interferons are going to be produced by two things. One, the white blood cells and the cells that were infected by the virus. So that's, I think that's pretty considerate of this cell to do that. As it is infected by a virus, it starts to release these interferons, all right, as kind of like a last ditch effort to kind of to try to save itself, but also to protect the cells that are close by. And in doing so, it helps to protect them from becoming infected. Because basically, when viruses infect a cell, they're basically going to turn that cell into a virus-making factory. They're going to use the parts of that cell, and it is going to use the resources of that cell to make more viruses. So what this does, what the alpha and beta interferons do, is it's going to help to destroy the nucleic acids there that viruses use to replicate, And it also helps to stop the virus from being able to make its own proteins. So these interferons stop that reproduction of the viruses. And then also what we'll see is natural killer cells will be called on hand to go in and, you guessed it, kill the virus infected cells. Now, gamma interferon is going to be produced by our T lymphocytes and our natural killer cells. And so they're going to call in the macrophages. Those are the heavy lifters. Macrophages do so much. And so they're going to come on in and kill the virus infected cells. So let's take a look at that. All right, so here's interferon release. So let's start up here with our infected cell. Okay, so here is our infected cell. Unfortunately, the virus has already gotten to it. It starts to release alpha and beta interferon in the surrounding cells, and so it protects the surrounding cells. And so these cells are going to then be stimulated to make, all right, certain proteins, enzymes, that are going to make it difficult for that virus to replicate. Now, meanwhile, the production of the alpha and beta gamma interferon, not gamma, alpha and beta interferon, right, is going to be released, and a natural killer cell comes across that and says, whoa, all right, it responds, and it travels over to the infected cell, all right, and it can do one of two things, okay? It can go to work on that infected cell, releasing, all right, its chemotoxic agents, right? The perforin and the granzymes, we saw how it kills those cells through apoptosis, but at the same time, it can release gamma interferon, which will then cause macrophages to migrate to that area. And those macrophages will just go in and phagocytize the whole cell. When they phagocytize the whole cell, they're basically going to gobble it up. And then the lysosomes inside of that macrophage are going to then go to work on that ingested cell. So again, think of this as the more the merrier. It's better to have too much than too little. Okay, we just don't want to have too much go on for too long. We draw out the immune response that could have some consequences. But again, we want our response to be short-lived. Okay, when everything's done and over with, we just go back to our normal operating procedure. Right, so it's like I said, it's better to have, all right, natural killer cells and macrophages being called to the area to scour the area, make sure that there isn't any infected cells remaining. Because when we get it all done, then we can clean up the mess and go home. All right, so still staying on that nonspecific internal defenses, we're going to talk about the complement system. Now, when I was in school, I had to memorize both the classical and the alternative pathway for the complement system. All right, and this is basically, all right, a whole series of reactions that occur, all right, from an immune response that involves many different proteins. Okay, many different proteins. All right, but we don't want you to, you don't have to worry about that. We only want you to be concerned with the end game. So what is the complement system? Okay, well, just like with anything, when you talk about, oh, hey, those shoes complement your outfit. Okay, they help out that outfit. They make it look nicer or more presentable. Okay, well, with our complement system, right, our complement system is going to work along our antibody system. All right, and so we call it, all right, when we're dealing with the complement system, you'll see a big letter C that we use, plus a number depending on which of those different plasma proteins that we're talking about. So C2, C4, C7, all of that fun stuff. Okay, again, you don't have to worry about it, but when you see it in literature, you see like C2, we're not talking about the second cervical vertebrae, all right, in your neck. We're talking about, all right, this is going to be one of those plasma proteins that are involved in our complement system pathway here, right? So where are our complement, where are these plasma proteins made? In the liver, okay? But they're released in the inactive form. as it should be, okay, because nothing's going on yet. So the release in the inactive form, they circulate around. It's like angiotensinogen, right? If you remember angiotensinogen, all right, the liver makes angiotensinogen, and it's an inactive protein, all right, that's in circulation, and it doesn't become activated until, all right, it gets to the kidneys, and then we'll see the creation of angiotensin 2, right, which helps deal with blood pressure issues. All right, but that was that chapter 19? Anyways, so when we're dealing with our complement, we produce it in the inactive form. And of course, like with most activation processes, it's going to involve an enzyme cascade, which means we'll activate, all right, certain proteins with an enzyme, and then it'll change into another type of enzyme, and then so forth and so on. So our complement activation, all right, depends on how that pathogen, okay, enters into what we're talking about here. All right, so you have the classical pathway. So we're going to see an antibody that is going to attach onto that antigen or foreign substance. And then the complement is going to bind onto the antibody. The alternative pathway, all right, we're going to be dealing with sugar, polysaccharide. It's either going to be part of the bacterial cell wall or a fungus cell wall. So complement is great when we're dealing with bacterial infections. So we're going to talk about the four different ways, all right, that we can activate, or I should say how we activate it, but how the effects of the activated complement are going to work here. So we have opsonization, inflammation, all right, cytolysis, and then of course, the elimination of the immune complexes here. So opsonization here, this is where we're going to see our complement protein, which is what we refer to as the opsin, is going to bind on to whatever that pathogen is going to be. And as a result of that, okay, we are going to undergo phagocytosis. So a quick picture here. Here you can see, here's your pathogen, okay? It's a bacteria. The complement binds onto that, all right? And that is going to attract one of our white blood cells, in this case, it's the macrophage, to come over and gobble the whole thing up. So it's kind of like when the complement binds onto the pathogen, it's like shooting a flare up in the middle of the night. And it signals, all right, one of our white blood cells to come over and to destroy whatever that complement is bound onto. All right, the next one is inflammation. Okay, a lot of stuff happens in inflammation, and we'll talk about that here in a moment, okay? But in this case, all right, what we're going to see is an enhanced inflammation, because what we're going to see is now we've activated our mast cells and basophils, and we know what they do. And at the same time, it's going to cause some chemotaxis, which is going to recruit neutrophils and macrophages on hand. So here you can see, all right, complement is going to be involved here with inflammation. So that's going to activate the mast cell and the basophils to release their substances, to get more cells to come to the area. And then we're going to attract more of the neutrophils and macrophages to that area. And after we get done talking about inflammation here in a few moments, you'll understand it a little bit better. But this helps to, okay, increase inflammation. All right, then cytolysis here. This is when I love this one. This one's really cool, because what happens is, all right, the complement is now going to cause a cell, all right, to have basically, it's like shooting something up with a bunch of holes. That's what we're going to see here, okay? So with cytolysis, complement's going to come over, and it's going to form, we used to call it the Big Mac attack, all right? But what the MAC is, it's the membrane attack complex, which basically we're going to just insert all these channels into the plasma membrane of that target cell. And what's going to happen? Things are going to flow into the cell until the cell literally bursts. That's what we see here. Okay, so here's your pathogen. All right, complement jumps on that and it forms the Big Mac attack here. And it forms these holes here into the pathogen, and then stuff will flow from outside the pathogen, inside the pathogen, and it causes it to burst and pop open. Pretty cool. And then last but not least, this is going to be the elimination of our immune complex. What is an immune complex? This is where we have those antigen-antibody complexes. But in this case, we're going to involve erythrocytes. The erythrocyte is like a mule. It's going to carry this whole thing. So what we're going to see is we're going to have these antibody-antigen complexes. So an antigen was identified. The antibody is going to bind with it, but now it's floating around. So the complement comes over and it's going to link these antigen-antibody complexes to erythrocytes. Then the erythrocytes are going to eventually wind up in either the liver and or the spleen. And what will happen in either one of those places is, all right, those antigen-antibody complexes are ripped right off of the erythrocyte, and then they're handled. They're taken care of. Then the erythrocyte can go back on its merry way. So that's what we're seeing here. Okay, so here's our antigen. Here's the antibody. So an antibody comes in contact with the antigen, it binds onto it. Well, then what happens is the complement comes over and says, all right, we got to get rid of this thing, clear it out of circulation. So then it jumps onto the antibody, then attaches itself onto our red blood cell. And it takes this whole thing is going to either wind up in the liver or the spleen, and then it'll tear this off, and then we'll take care of that antigen, usually through one of our white blood cells, gobble it up or whatever. So let's talk about inflammation. Okay, so that's complement now. You see how the complement is just going to kind of enhance the immune system, helps through certain processes. I'm telling you, your immune system is such a cool system in the body. There's so much that goes on there that you don't even realize, right? And so the complement is just another facet of the immune system. Wait till we get to the acquired immunity. That's going to be much more interesting, at least in my mind. All right, so let's talk about inflammation, because it's important that we talk about inflammation here. All right, so when we're dealing with inflammation, all right, something has occurred, usually let's say through an injury. Okay, you've scraped your leg, okay, and some foreign antigens, some foreign pathogens like bacteria, viruses have gotten into, all right, your tissue. So basically, inflammation is going to be that process that we're going to, that the body's going to create to basically get rid of these unwanted substances. Okay, so one, it's local. Okay, it won't be widespread. You know, if you scrape your knee, you shouldn't have this process going on at the back of your head. All right, it's also nonspecific. It's local and nonspecific, and we have to have blood vessels involved. And finally, this is part of innate immunity. All right, so let's walk through the events of inflammation. Now, we talked a little bit about this in chapter 6, all right, when we talked about the healing process of the skin. We didn't really focus on the events of inflammation because that occurs, all right, that's part of the healing process, right? But now we're going to get into the specifics of inflammation. So in order to have inflammation, you have to have injured tissue. We have to, okay? So here's what happens. You damage the tissue, the injured tissue, all right, the basophils, which are going to be in the in the vascularized portion of that tissue, the mast cells, which are sitting outside of those blood vessels in that tissue, and any infectious organism, virus, okay, a fungus, a bacteria that is infiltrated and gotten into your tissue there, all of those are going to release chemicals. That is important. So we have all these different things releasing chemicals. So the injured tissue is going to release the chemicals, the cells there, the basophils, the mast cells, and even the infectious organism that came in, all right, because of this injury, are all going to release chemicals. And this is going to start the ball rolling. All right, so we already talked about, right, what chemicals are going to be released. Histamine, all right, mast cells and basophils are going to release the histamine to make the blood vessels leaky. All right, leukotrienes, prostaglandins, those are part of those eicosanoids that I was telling you about. All right, so those eicosanoids, those locally acting hormones that are going to come from damaged tissue, all right, which are going to act, all right, in the inflammatory process, okay? But also we're going to see what we call those chemotactic factors, or those chemicals are going to attract our white blood cells to that area there. All right, so let's start from the beginning here. All right, so we've damaged the tissue. So next, we're going to see, all right, the blood vessels that are in that damaged tissue undergo some changes. One is vasodilation, okay? We make the lumen of the blood vessels bigger, but in doing so, we have now made our capillaries leaky. So the blood and, well, not the blood, but the cells that are in the blood and some of those proteins can now leak out into the tissue. Now we've got a new thing on hand, what we call CAMs, stand for cell adhesion molecules. And so these CAMs are going to appear on the endothelium. And in case you forgot what the endothelium is, that is the name that we give simple squamous epithelial tissue that lines vessels. That includes blood vessels and lymphatic vessels. So we're going to see these CAMs start to appear, and they're going to grab onto the leukocytes. I would think of it like this. If you ever look at a river, okay, if you watch the water flowing down the river, right, the water that flows down the center of the river moves much faster than the water at the edge. Okay, and the same thing applies for blood flow. We talked about this before. The blood in the center of the blood vessel, all right, undergoes what we call laminar flow, goes straight down. But the blood on the periphery, all right, is slower, and it creates what we call eddy currents. All right, so what we're going now, imagine somebody falling into a river, and they're in the middle of the river and they're floating down, all right? And so we're like, oh my gosh, there's a little kid that fell in the river. We've got to try to save them. And it's in the middle of a festival that happens to be at this river. And there's people that run to the edge of the river, and they stand on the riverbank and they put their hands out, okay? That person that's in the river is going to be a white blood cell. So if they come close to the edge of the blood vessel or the riverbank, people reaching out their hand, they're going to try to grab onto that person that's in the river. Well, that's what these CAMs do. They are going to make it where the leukocyte can then stick onto, all right, the edge of the capillary there. Once that happens, when we get the leukocyte to adhere, all right, we call that process margination. And margination is basically on the margin, the edge of the blood vessel, in which the leukocytes are going to stick onto the endothelial cells. Once that happens, now we're going to try to get that white blood cell to leave our bloodstream and get into the tissue. That process is called diapedesis. It will literally squeeze in between the cells of the blood vessel walls. And they are going, how do they know where to go? No problem, through the process of chemotaxis. Because remember all those structures that I told you that were releasing chemicals as a result of inflammation of that tissue that was damaged, okay? It is going to migrate toward the source of those chemicals, which is going to be any of those cells that were damaged in the tissue. those pathogenic cells, all right, that bacteria or virus that's there, all right, and any of the white blood cells that were already on the site immediately. So while this is going on, okay, we're going to cause a migration of white blood cells to the area. Well, we probably need to make more. So we are going to see an increase in leukophoresis in your red bone marrow. How do we do that? Through the release of cytokines. And then macrophages are going to release what we call pyrogens. And they are going to cause fever. Actually, fever is good. It really is. I mean, a really high fever is not. 109 is bad. Okay, 106, not very good. Okay, but if you have a fever of 100, that's not bad. Okay, you might not feel good, but point being is your body is trying to make it a hostile environment because bacteria will like to reproduce, right? But they want to reproduce in a comfortable environment. So if your body turns the temperature up, it's not so comfortable. So it's going to make it more difficult for them to reproduce. Okay. So meanwhile, we're getting the cells. They're trying to leave the bloodstream to get to the area of injury. Now, we're also, don't forget, what else is in our blood? Okay. Well, we have our plasma proteins. Our plasma proteins are made up of, all right, tons of albumin, okay, but that's not really so important here, but immunoglobulins, what are those? Antibodies, okay? So we have immunoglobulins, complement, which we just got done talking about, our clotting proteins, which we've learned about, because eventually we've got to clot this up, right? And we'll talk about that in a second here, and then kinins, which we'll talk about here in a second. Of course, our clotting proteins are going to form clots, but yes, it's going to help to stop the blood flow, but it's important because it walls off the microbes. We don't want those microbes getting into circulation because then they can go anywhere. That's not good. We don't want that. So we want to wall them off. We want to contain this infiltration here. Okay, so we got our clotting proteins, and they're going to start clotting. They're going to start to clot and prevent the entry of any more microbes from the external environment. Okay, we got to seal off the external environment. It's like in a sci-fi movie, if there's a hole in the side of a spaceship, all right, what do they try to do? They try to stop, all right, that hole, all right, from causing any more issues. So they try to close it off. All right, our kinins do a couple different things. One, they're going to stimulate the pain receptors. Okay, bradykinin plays a huge role in pain response, right? But also they're going to help to increase how leaky our capillaries are, and they're going to help to make more CAMs so we can get more leukocytes that can leave our bloodstream and get into the tissue. Right, so these proteins are made, okay, in the liver and some other cells, but they're made in the inactive form, and they'll be activated. And that's that bradykinin, which I was telling you about, big time in pain response. All right, so my son had a blister on his foot when we went to Universal Studios recently. And it hadn't popped, and so he was curious about it. And so I got a chance to talk to my son, who's 14, and we discussed what exudate was. And he actually found it to be quite interesting. But what is what we call our fluid exudate? And we talked about how in our blood, okay, we have fluid in there besides the blood-formed elements. Well, the plasma, right, majority of it's made up by water. That's true, okay? But we then have a bunch of our proteins in here. So this fluid, or we call exudate, okay, we want it to go from the blood to the injured or infected area, right? Because what is in this exudate, right? Yes, it's a lot of the blood plasma fluid, which is going to be a lot of water, but also those proteins I was mentioning, but also our immune cells. And those proteins will help with the healing process. So that is one of the reasons why we see vasodilation. When we get an increase in the lumen of the blood vessels, we're going to get an increase in blood flow to that area, which is what we want. So now we have an increase in blood flow, so we can get more of the stuff that we need, okay? And then we're going to make it leaky because, all right, we have these gaps that open up in between the simple squamous epithelium there, the endothelial cells. So that gives us more, so now we can increase that leakiness. And then don't forget about our plasma proteins. They exert that capillary osmotic pressure. Remember that oncotic pressure that we were talking about that was inside the capillaries? Well, if our blood vessels become leaky, we're going to lose plasma proteins. Where are those proteins going to go? They're going to go out into the tissue, and they're going to take that attractiveness, all right, for fluid with it. So we're going to decrease the osmotic pressure inside the capillary, right? And so if we decrease that osmotic pressure, right, then we're going to see a decrease in fluid reabsorption into the blood, which means more fluid will flow from the blood vessel out into the tissue. Remember bulk flow. If you're not sure, go review bulk flow, right, and how we calculate hydrostatic pressure and how we calculate our oncotic osmotic pressure or colloid pressure. Okay, so then as that fluid washes out into the tissue, okay, what we'll see is this process called washing. And that extra fluid, all right, that washes out into the tissues will be absorbed or taken up by the lymphatic capillaries. All right, a lot of it will help to be taken up by the capillaries themselves, the blood capillaries, but roughly about 15%, all right, that extra fluid that's not taken up, the lymphatic capillaries will help to take that up. And so think of that as kind of like the cleanup crew. They're going to take away any debris, all right, and any of the other stuff that resulted, all right, from that injury there, and we whisk it away. So this whole process takes about 72 hours. Okay. So during this time, all right, as we achieve our goals, all right, of forming a blood clot, getting enough leukocytes into that area to help with damaged tissue, taking care of pathogens, all right, those macrophages go in there. They start to eat away at the bacteria, okay? They will come over and chew up, all right, any of that damaged tissue and get rid of that. And we have to get rid of our dying neutrophils because they've been very busy going over there and destroying any of the bacteria there. And then we talked about this in chapter 6, our fibroblasts show up on the scene and they're going to lay down collagen and help to repair and fix and make that new connective tissue. And hopefully we'll see regeneration instead of fibrosis. We don't want scar tissue, all right? We want to get new tissue in that area there. So here you can see our friend here, the skateboarder, who has fallen and skinned their knee here. And as a result, right, some bacteria has infiltrated the area. Okay, you see some redness here from increased blood flow, but the bacteria now have entered into this area. And so they're releasing, all right, chemotactic factors, and the injured tissue is going to be releasing chemotactic factors, right? And that is going to cause any of the leukocytes in the tissue here to be attracted to that area right away. And as that's going on, all right, we have our mast cells that are close to the blood vessels. They're going to be releasing histamine and heparin. And so that histamine and heparin, all right, is going to help, all right, create vasodilation, making our blood vessels leaky. The basophils in the blood will be doing the same thing. And so we'll start to see, here's those little cams here, all right, that are going to be displayed on the endothelial cells. And so the neutrophils and other leukocytes that are rolling through here, all right, well, when they bump up against these cams, they'll start to stick to the wall here, right? And we call that margination. So it'll slow down these cells long enough where they can start to squeeze through. You can see these gaps that are appearing through that vasodilation there, all right? And through those chemical substances that are going to make our blood vessels more leaky. Now the cells start to squeeze through, and we call that diapedesis. Then once they've squeezed through, they're going to be attracted to the area of injury because of all of those items that I've discussed that are releasing, all right, their chemotactic factors. And these cells are going to migrate to that area and start attacking, okay, the bacteria or any other pathogen that's in that region. And again, we are seeing the leakiness of these blood vessels. Plasma proteins are going to start to leave. And that's going to facilitate more fluid leaving the blood and going into, all right, the tissue here. And we call that washing, all right? And so that washing is going to flood that area, all right, with that exudate. All right, and then anything that's not getting absorbed by the capillaries, we have our lymphatic capillaries that will take up that excess fluid there to kind of help drain the area. Again, it goes into the lymphatic system, and we'll talk about the lymphatic system in another chapter, right? But eventually that gets dumped back into the circulation. So we have a lot of different ways to kind of clean up this mess after it occurs. So what do we see, all right, for inflammation? Okay, well, in the picture there, you saw redness right around the area here, all that redness right there. Okay, that's from an increase in blood flow. Heat, all right, is because we're going to see an increase in metabolic activity. All those cells are working hard when we're trying to fix damaged tissue, all right? You have the white blood cells doing their thing, right? So most reactions when we're undergoing metabolism or metabolic functions, we're going to have what we call exothermic reactions, which is giving off a heat for a lot of these chemical reactions. So we'll see an increase in heat there, but also an increase in blood flow to the area. Blood is 1 degree warmer than our normal body temperature. All right, swelling. Well, we already know about that increase in fluid, all right, that comes into the area because those plasma proteins are leaving the area. And so that's going to cause more fluid to leave the capillaries and move into the tissue there. And then pain, right? We have all those pain receptors. If you remember from chapter 16, pain receptors are going to be non-encapsulated or unencapsulated receptors. And so those receptors are very, very sensitive. And so a couple stimuli that will cause those pain receptors to be activated is one, pressure. All right, the compression of that extra fluid in the area is going to cause pain, but also chemical irritants like bradykinin, okay? And then we'll see a loss of function, all right, because if we have a lot of inflammation, right? And you probably, I don't know if you've ever had that experience where you may have sprained your ankle. I had a 2nd degree ankle sprain, which is just short of putting me in the hospital, but my ankle was swollen like no tomorrow. And when eventually the swelling went down, there was tons of black and blue bruising there. And again, that's because a lot of the blood vessel, the erythrocytes were damaged from that process. But I had quite a bit of swelling, which if you increase the swelling a lot, you'll definitely increase the pain. And the function that I lost, because it was an ankle joint, I just did not have much mobility or movement there. All right, so normally when we talk about acute inflammation, it's going to be a short period of time, anywhere between 8 to 10 days. Chronic inflammation, all right, will be much longer. And yes, it does have detrimental effects because chronic inflammation can eventually lead to tissue destruction, all right? And especially if you're dealing with joints, if you get tissue destruction, you're going to get degeneration, and eventually you're going to get, I hate to say it, but arthritis. All right, so what are some of our non-specific, all right, occurrences here when we're dealing with, all right, innate immunity? Fever, okay? You'll notice our body temperature increases outside of that normal range, all right, of 96.4 to 99.1, go up to 102, right? So if we see a degree 1 centigrade, not Fahrenheit, but 1 degree centigrade or more, that's considered to be a fever. And that's caused by what we call pyrogens. And those are going to be, all right, these chemicals, all right, that are going to be released either from whatever it is that infected you, or if your immune cells, right, and what it's going to do to affect your fever, your body temperature, is based on how the hypothalamus, okay, remember the master gland, as some people refer to it, it's all part of the diencephalon up in the brain there, chapter 13, all right, you will see the effects. We're going to talk about that here in a second. An example that's given here is interleukin 1, all right, is one form of the pyrogens here. So these cells, for example, a macrophage, right, can release pyrogens. And so that macrophage releases some pyrogens into circulation, and it eventually winds up at the hypothalamus. And so that pyrogen causes the hypothalamus to release prostaglandin E. This is another eicosanoid, okay? But prostaglandin E2 is going to cause an increase in your body temperature, causing the fever. Okay, and we'll talk about the effects, why fever is good and bad here in a second. Almost done. All right, so fever has 3 stages here. You have onset, stadium, and deferescence. So onset, obviously, is when your body temperature starts to increase. And so what we'll see is the hypothalamus is going to cause vasoconstriction. And that's going to cause vasoconstriction in those blood vessels found in your skin to help, all right, retain the heat. Because again, all right, these pathogens like to have a certain temperature. If we move them outside their comfort zone, it will impair their ability to reproduce. So if we want to increase our body temperature even more, let's get some shivering going on. Get those skeletal muscles to relax and contract and generate more heat. Okay, so this is what you get, the chills there. So now we're starting to increase our body temperature. Now for a while, we're going to maintain that body temperature. All right, we've seen an increase in metabolism of those cells there, okay, as the white blood cells are working overtime to try to destroy and get rid of any bacteria or viruses that have gotten into your body, all right? And the other cells, if there's a damaged tissue there going on, okay, well, those cells are going to be working overtime to try to repair the area. Interesting fact, the liver and the spleen are going to withhold, all right, zinc and iron, which are 2 substances that these microbes need to make more of themselves for reproduction. So it withholds the zinc and the iron, therefore there's less material to reproduce, making it harder to reproduce. And then finally, all right, we've got the deferescence there. When we return our body temperature back to normal, okay, the hypothalamus is no longer going to be stimulated by the pyrogens, all right, from the cells and the infectious cells there, all right, they're no longer being produced. So then the hypothalamus stops producing prostaglandin E2, all right, and then we will switch from vasoconstriction to vasodilation of those blood vessels in the skin to let more blood flow, all right, to get rid of that heat, all right, and then you will even start to sweat, and that's usually a good indicator. You've heard people say, oh, I broke the fever. I broke the fever because I'm sweating. Okay, why is the fever good? Well, like I said, it makes it hard for bacteria and viruses to reproduce. Okay? It also helps with some of our chemical substances, the interferons, all right, for them to do what they need to do. We haven't talked about adaptive immunity, but we will. But this helps to increase adaptive immunity, right? Helps to increase tissue repair, increases the amount of CAMs that we see on the endothelium, which is great. And of course, all right, like I said before a few moments ago, all right, if you have a low-grade fever, that's okay. Most times, all right, it's encouraged that you don't do anything for that low-grade fever. And there was, if you talk to some of your medical professionals, for those of you that have ever gotten the COVID vaccination, they said for people that if they do spike a fever, all right, if it's a low-grade fever, don't take any like Tylenol or ibuprofen, anything to reduce the fever. Unless it gets out of control, then yes. But they said that if you do that, you can, you can impair, all right, the immune response. Specifically, you can impair the adaptive immunity response. And you don't want to do that, all right, unless the fever gets too high. Well, what's a high fever? Well, let's look at it, all right? Yes, it can be potentially dangerous here, all right? And big time, because it can change, all right, how tissues undergo certain metabolic pathways. But really, what we want to make sure is we don't want your proteins to undergo denaturation. That's basically when the proteins start to unravel or they unfold, right? And when that occurs, then the proteins can no longer function. And if you know the functions of proteins, there's many functions for proteins, all right? Obviously, protection is one through antibodies. Channels in certain cells can no longer function. Big problem there. So we have to be careful of that, all right? When the temperature gets really high, you can have seizures. That's bad. So when you get up to like 103, 104, things aren't so good. You get up to 106, you can have brain damage. And then you get up to 109, you can die. Got to be careful with that. So after an injury, you may see, all right, pus, which is another form of exudate. All right, now pus is going to be a little bit thicker than the type of exudate that you saw with the blister. All right, that's more of what we call a serous exudate, which is more liquidy. All right, pus exudate, this one's going to be thicker because you're going to have, all right, dead viruses, bacteria, whatever. You're going to have the fallen soldiers here of our white blood cells, the macrophages, and then any cellular debris from the tissue that may have been damaged from whatever occurred. All right, now a lot of this pus and exudate is going to be removed, all right, through the skin. You might see it like, for example, a pimple, all right, or something a little bit larger, or the lymphatic system comes in and tries to clear things up. Now, if it doesn't get cleared all the way, then you could form an abscess. For those of you that like to watch that show, Dr. Pimple Popper, I refuse to watch it, okay, in which this person goes in and takes care of all these abscesses, all right? You can get that to occur. And basically, it's the body just saying, hey, all right, I'm going to kind of wall off that pus with collagen. And in some cases there, right, we just need the removal of that collagen, that pus, through surgical intervention there. So what's the deal with chronic inflammation? Chronic inflammation lasts longer than 2 weeks, right? And this is when, again, we need to have a point in which we need to shut things off. I'm not going to leave my car running all night, all right? It's not good for the car. It's not good for the environment, what have you, all right? So when we're dealing with chronic inflammation, we're going to see, all right, our macrophages and our lymphocytes, they're going to be on hand, and they're going to be constantly doing what those cells do, all right? Problem with that is, all right, like I just showed you on this page, all right, we are going to end up with dead leukocytes, dead macrophages. So then that's going to constantly be causing leukopoiesis to occur in your bone marrow, right? And so what we want to do, we don't want to be wasteful. We don't want these cells to just go out of sight here, So we see this quite a bit with our overuse injuries. And what we need to do is, all right, we need to shut this down. And so if our acute inflammation can't do that, then chronic inflammation can become problematic because we are wasting resources. Now, chronic inflammation could be caused from a variety of reasons. One of the ones that we get concerned about is if it's autoimmune, where your body just kind of attacks itself. Those white blood cells start to go a little bit out of control and they can't recognize self from non-self. And that's where we have to be concerned about. And I mentioned before how if chronic inflammation goes on untreated, you can get tissue destruction. Right? And if you get tissue destruction, you can get loss of function. And if you get loss of function, you can get scar tissue formation. If you get scar tissue formation, that's going to be an issue. All right, folks, that's going to end it here. I know I went a little bit long today, but I really wanted to cover all of that information. And I'm just talking to myself. So that's fun.